All right, Mr. Martin's IB biology class. We are diving deep into DNA replication today.
Yeah, that's right.
I'm your host.
And I'm the expert here to break it all down.
Now, DNA replication.
It can seem pretty complex at first glance.
Yeah, but we're going to show you that it's actually super elegant.
Absolutely, and really essential for life.
So what exactly are we talking about when we say DNA replication?
Well, in the simplest terms, it's how a cell makes a perfect copy of its DNA.
It's like hitting Control-C and Control-V on your computer.
Exactly, except it's happening at a molecular level inside every living cell.
So why is this so important?
Well, think about it for a second. Every time a cell divides,
It needs to give a full set of instructions to each of its daughter cells.
and those instructions are encoded in the DNA.
So without replication,
Wouldn't have growth,
you wouldn't be able to repair damaged tissues.
and passing on genetic information to the next generation would be impossible.
From tiny bacteria to giant redwood trees.
DNA replication is the foundation of life as we know it,
Now we're mainly going to be talking about the process in eukaryotic cells.
which are the cells that make up you and me.
But it's important to note that the general principles are pretty much the same across all forms of life.
So whether you're talking about bacteria, plants, or animals.
The basic mechanism of DNA replication is remarkably conserved.
And that's what we're going to be exploring today.
The key steps, the molecular players.
And even some cool real-world applications of this knowledge.
So let's imagine for a second that we're looking at a single human cell.
Under a super powerful microscope, of course.
Inside that cell, we'd see a nucleus,
That's where the DNA is stored.
like a library full of genetic information.
And that information tells the cell how to function.
It's basically the instruction manual for building and running an organism.
Now, when a cell gets ready to divide,
It needs to make sure that each of its daughter cells gets a complete copy of that instruction manual.
it can't just rip the manual in half.
Right, it has to make a whole new copy from scratch.
And that's where DNA replication comes in.
So we're talking about duplicating an entire molecule of DNA.
Which can be billions of base pairs long.
It's a mind-bogglingly complex process.
But the cell does it with incredible speed and accuracy.
So how does it actually work?
Well, the first thing to understand is that DNA replication is what we call semi-conservative.
Semi-conservative, what does that even mean?
It means that each new DNA molecule is made up of one old strand
From the original DNA molecule.
and one brand new strand.
So it's like taking a zipper and unzipping it.
And then using each half of the zipper as a template to build a new zipper,
So you end up with two zippers.
each with one old side and one new side.
That's a great analogy.
And the semi-conservative nature of replication.
Is one of the reasons why it's so accurate.
Because each new strand is built using an old strand as a guide.
So let's zoom in even closer and look at the individual building blocks of DNA.
Those building blocks are called nucleotides,
And each nucleotide is made up of a sugar
a phosphate group,
and one of 4 nitrogenous bases.
adenine, thymine, cytosine, and guanine.
You'll often see those abbreviated as A, T, C, and G.
And the key to DNA replication is that these bases pair up in a very specific way.
A always pairs with T,
And C always pairs with G.
like puzzle pieces that only fit together in one way.
And these base pairs are held together by weak bonds called hydrogen bonds.
So when the DNA molecule unzips,
Free nucleotides floating around in the cell can come in and pair up with the exposed bases on each template strand.
following the strict base pairing rules,
So if there's an A on the template strand,
only a T nucleotide can pair up with it.
and if there's a C on the template strand,
Only a G nucleotide can pair up with it.
so the sequence of bases on the old strand dictates the sequence of bases on the new strand.
And this ensures that the two new DNA molecules are exact copies of the original.
Now, this whole process doesn't just happen spontaneously,
There are a bunch of enzymes involved.
each with a specific job to do.
Kind of like a construction crew working together to build a house.
One of the most important enzymes is called helicase.
Helicase is like the demolition crew.
It comes in and breaks the hydrogen bonds that hold the two strands of DNA together,
Essentially unzipping the DNA molecule.
creating a replication fork where the two strands separate.
Now, once the DNA is unzipped,
Another enzyme called DNA polymerase comes into play.
DNA polymerase is like the construction worker that actually builds the new DNA strands
It reads the sequence of bases on the template strand,
and adds the complementary nucleotides to the growing new strand,
one by one,
following those base pairing rules that we talked about,
A with T,
and C with G.
and DNA telomerase is a very efficient worker
It can add thousands of nucleotides per second.
with amazing accuracy.
And there's one more important thing to mention about DNA polymerase.
It can only add nucleotides in one direction.
From the 5' end of the new strand to the 3' end.
So one strand, the leading strand, can be synthesized continuously.
But the other strand, the lagging strand,
Has to be synthesized in short fragments called Okazaki fragments.
because DNA polymerase has to keep jumping back and starting again
And then another enzyme called DNA ligase comes in
and joins those fragments together.
to create a continuous new strand,
So it's a pretty complex dance of enzymes.
all working together to make sure that DNA replication happens quickly and accurately.
Now, I know we've been talking a lot about the nitty-gritty details of replication.
But it's important to remember why this process is so important in the bigger picture.
Without DNA replication,
Life as we know it wouldn't exist.
cells wouldn't be able to divide,
Organisms wouldn't be able to grow.
and genetic information couldn't be passed on to future generations.
So it's really one of the most fundamental processes in biology.
And speaking of applications,
This knowledge of DNA replication isn't just theoretical.
scientists use it all the time in the lab.
For things like DNA fingerprinting,
Genetic testing.
and even developing new drugs and therapies.
One of the most important techniques that relies on DNA replication is called PCR.
Which stands for Polymerase Chain reaction.
And as the name suggests,
It uses DNA polymerase to make lots and lots of copies of a specific DNA sequence.
it's like a molecular photocopier,
And it's incredibly useful for a wide range of applications.
like forensic science
medical diagnostics.
and even evolutionary biology.
So how does PCR actually work?
Well, it's basically a three-step process.
That's repeated over and over again.
The first step is called denaturation,
And that involves heating up the DNA sample to separate the two strands.
just like helicase does during replication.
But in PCR,
We use heat instead of an enzyme,
the second step is called annealing.
and that involves adding short pieces of DNA called primers to the sample.
These primers are designed to bind to specific sequences on the target DNA.
So they basically tell DNA polymerase where to start copying,
And the third step is called extension
and that's where DNA polymerase comes in,
and adds nucleotides to the primers.
to create new DNA strands that are complementary to the original template.
And since each cycle of PCR doubles the amount of DNA,
You can end up with millions or even billions of copies of your target sequence after just a few hours.
it's really remarkable.
And PCR has revolutionized many fields of biology.
It's used to diagnose genetic diseases
Identify criminals.
and even study ancient DNA.
So understanding DNA replication,
Isn't just about memorizing a bunch of facts for a test.
it's about understanding a fundamental process that underpins all of life
And it's about appreciating the incredible power of molecular biology.
to solve real-world problems.
All right, Mr. Martin's class, we hope you found this deep dive helpful.
We've covered a lot of ground today.
But we've only just scratched the surface of this fascinating topic
There's so much more to learn about DNA replication.
and its implications for everything from human health to the evolution of life on Earth.
So keep exploring, keep asking questions.
And remember that the world of biology is full of wonders waiting to be discovered.
If you enjoyed this episode, be sure to like, comment, and subscribe to our channel for more deep dives into the world of biology.
We'll see you next time.
Bye for now.